This is a production of Cornell University.
Okay, I think the numbers have stabilized. Many of you already know me. I'm Lucia Viñale. I'm organizing our lab's annual hemp webinar. I am a third-year PhD candidate in the Larry Smart Lab at Cornell University, and I'm working on hemp powder mildew in hemp. We are focusing on hemp grain this year, and today's speaker is Ian Graham, I'm sorry, who is joining us from the UK. Thank you, Ian, for accepting our invitation. We're all excited to hear your talk. Following postdoctoral fellowships at the University of Oxford and Carnegie Department Plant Biology in Stanford, California, Ian took a faculty position and set up his own lab in the UK at the University of Glasgow in 1994, when I was born. In 1999, he moved to become the Weston Chair of Biochemical Genetics in the Center for Novel Agricultural Products at the University of York, where he remains with his research focused on biosynthesis of plant natural products and how these can be developed for various industrial applications. Representing the University of York, Ian was granted the UK plant breeders' right for a high oleic hemp seed variety in February of 2021. Ian was elected a Fellow of the Royal Society in 2016 and is a Science Trustee of the Royal Botanical Gardens. Okay. Ian is also the academic lead for Yorkshire, a partnership involving the University of York, Ferroscience, and Askham Bryan College that aims to deliver green economic growth, and is the director for the UK government-funded high-value biorenewables network. The title of his talk is Molecular Breeding of New Seed Oil Varieties of Industrial Hemp. The webinars are recorded and uploaded to our School of Integrative Plant Science, SIPS YouTube channel. Feel free to check the 4 previous ones that we have already uploaded to our channel. And with that, the virtual stage is all yours, Ian.
Good. Yes, thank you. I should first of all apologize for providing you with such a long bio. It's taken about 5 minutes of our valuable time. So it's a great pleasure to have had this invitation from Lucia and the people at Cornell. I think it's a great idea to have it such a global seminar. I honestly didn't appreciate it was so global, but welcome everyone, and thank you for turning up to listen to me for the next 40 or 50 minutes. This is a story which is really close to my heart. I've been working on hemp in the background of my other main projects on medicinal plants for the last 15 years almost. And I think, as in other places, getting funding to work on hemp has never been all that easy. It's maybe getting a little bit easier now. With the relaxing regulations around planting and growing and all of that, especially in North America. But yeah, it's a great opportunity to tell you about some of the work that we've done, and also maybe give you some insight into seed biology, which is another one of my interests and has been since I started my lab the year that Lucia was born, as she pointed out. So that kind of makes me feel a bit old, but there you go. So molecular breeding of new seed oil varieties of industrial hemp. I think industrial hemp, when you talk about it, most people think about fiber, but my focus and my interests over the last 40 odd years have been very much on seeds and oil, and then progressing into other exotic medicinal crops. The concept which I'm going to introduce, and many of you will be familiar with in terms of altering traits in industrial hemp varieties, is reverse genetics. And that's really going from gene to phenotype rather than forward genetics, which is going from phenotype to gene, as it were. And of course, the potential of reverse genetics in orphan crops being those that are not all that well studied or being developed by molecular breeding tools in particular, The whole revolution in DNA sequencing and knowledge of the genome and genome assemblies now as well are greatly facilitate this concept of reverse genetics, because we can get to so-called candidate genes much easier than we ever could before. And I'll give you some insight into how we went about doing this even before the sort of that revolution had really taken off. So first of all, just let's talk a little bit about industrial hemp. It's of course a multi-purpose crop. We use that terminology, industrial hemp, for low cannabinoid hemp that's grown mainly for fiber and seed and then from seed oil applications, as you'll see. Uh, and as you'll see in my talk, uh, that whole concept of doing transcriptome analysis has allowed us to identify genes and modify genes involved in the seed oil composition. And in North America, in fact, in Canada, industrial hemp is still grown largely for seed rather than fiber, and there's huge potential to develop this crop as a multi-purpose crop for fiber as well as seed. And maybe I'll just say initially that hemp has been grown a very long time, and in fact in the UK during the reign of Henry VIII, when in the 1600s, every farmer in the country was legally obliged to grow a rood, which I think is about a third of an acre of hemp, because it was so much in need for sailing ships, for the sails and for the rope of those sailing ships. So you can see it's been a very important part of of the UK as well, but it's fallen right back, and my ambition is to really increase the acreage of it again from the something like 800 hectares that are grown mainly for fiber in the UK today. So yeah. So just a little bit then about the crop. I don't want to dwell on this because those of you who've tuned in, you're interested in hemp, you'll know all of this. But it's an annual herbaceous plant. I wanted to make the point that you get both monoecious and dioecious varieties. Dioecious varieties are those where the male and female flowers are on separate plants, in fact. And that introduces challenges itself to breeding programs. So it basically behaves like separate male and female individuals for crossing, and I've mentioned the different drug types. It originated in Europe, northern India and Persia, but it's now cultivated around the world in warm to temperate regions, and 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 there are good claims around it being used for low input farming. And in fact, in the UK, one of my ambitions is to really get it adopted as a break crop to grow on good arable land where cereals are growing, where every 3 or 4 years you need to have a break. And at the minute, that's large. That break crop is often oilseed rape or canola, as you refer to it in North America. And but there's all sorts of issues with that as being really terms of, in fact, in Europe now, the main sprays that are used in canola are being banned, and so they are looking for alternative break crops, and industrial hemp is one such that could really be adopted, and farmers like growing it. And so the applications are in fiber and cannabinoids and in seed oil. The industrial hemp varieties they need to be below 0.2% THC in the UK for us to get a license to grow them. I think in the other places it's 0.3%. And now if we just take a look at this seed, it's got about 30 to 35% oil, 25% protein. The amino acid composition is really good for human health. And if you go into health food shops, you can often buy hemp seed powder because of the protein composition. And the oil, as we'll come on to, is also very interesting. Good for dietary fiber, vitamins, and minerals. So nutritionally, it's very interesting and is usually in those high-end nutritious foods. So just as when we started this project, and in fact still in North America, this variety, Finola, is one of the favorites that's grown in Canada in particular. There are other options as well, of course, but Finola itself is interesting. It's a relatively dwarf variety, which means that you can combine harvest it for seed. Early flowering at 25 to 30 days, early maturing at 115 days, and it's a really nice diploid variety that we really like to grow and work with here in York as well, because it's fast cycling, so we can get a couple of generations in a year, for example, under glass, and can be grown at high latitudes, hence its popularity in Canada. Good rotation crop. And I put in here a seed yield of 1 to 2 hectares. There's claims out there that you can get above 2 hectares, and it's like any crop, it's not magic, and what you get out what you put in, and so it depends on the soil and the conditions for growth. I'll say something about the actual fatty acid composition, because it's the fatty acid composition in the seed oil that makes it interesting, in particular for hemp. And so what we've got here is the different types of fatty acids along the top, and then I've compared hemp this table compares hemp with other oil crops, as it were. And you can see immediately that hemp has got high levels of linoleic acid, which is 18 carbons, 2 double bonds. That's what this numbering represents. And alpha linolenic acid, which is 18 carbons, 3 double bonds. We'll come on to this in my biochemistry tutorial in a minute. And then the interesting one that I'll be focusing on is oleic acid, which is 18 carbons, 1 double bond. And so you'll see that the number of double bonds increases as we go across this table. And this PUFA is polyunsaturated fatty acids. So you can see that there is and polyunsaturated is anything more than 1 double bond. So you can see that the level of PUFAs in hemp is high at 84%. And if you compare that with olive oil, then you can see immediately that olive oil has got an elevated levels of oleic acid, 18 carbons, 1 double bond, and then all of these other ones. Nutritionally and health-wise, both alpha linolenic acid, which has got 3 double bonds, and gamma linolenic acid, we'll come onto these in a minute. You can see that many of these oil crops don't have any GLA. Hemp is relatively special, let's say, rather than unique, in having 4% GLA, and this ALA GLA, which is omega-3 and omega-6 oils, again, many of you will be aware and may indeed take them as supplements, are important for your health. And so we were interested in this when we thought about changing the composition, because obviously if you compare the levels of GLA, gamma linolenic acid, to borage, which is very well known to have elevated levels of GLA, it would be nice to have more GLA in hemp oil, obviously. But one of the issues with polyunsaturated fatty acids is that they get they are oxidized. They become oxidized as soon as you take them out of the seed and in that protective capsule. And in fact, just like fish oil, which has got elevated levels of polyunsaturated fatty acids, in fact, 20 carbon and 22 carbon fatty acids in fish oil. But that smell of fish oil that you get when the fish isn't fresh is because the fish oil is becoming oxidized. And exactly the same happens, in fact, with hemp oil. When you take it and leave it open to the air, it becomes rancid. And so because of that, it's not really all that attractive for cooking or for processing, for processed foods. And so obviously then one of the targets could be, how could you make this fatty acid composition more like olive oil and increase the amount of oleic acid? And in so doing, you would predict increase the stability. And so that was sort of the context that we came into this project to see if we could do something about that. And then I just want to then summarize very quickly what happens in a developing seed when fatty acids are being made that accumulate in the seed to give that vegetable oil that we get from all plant seeds, in fact, and also, in fact, from olive oil, which of course isn't derived from seeds. And so we've got here, this is the plastid, which most of you will be aware of, that's present, and this is involved largely in photosynthesis, but it's also involved in that first stage of fatty acid biosynthesis in seeds. And in fact, fatty acids are synthesized in developing seeds and in seeds all around the plant up until they reach that 18 carbon 1 double bond oleic acid that I mentioned. And this is a fatty acid desaturase that's soluble, and it's quite unique in just having this job of converting 16,0 to 16,1 palmitoleic acid and stearic acid to 18,1. And then the fatty acids come out into the cytosol, and there is this whole suite of fatty acid desaturases then that keep introducing more desaturation and double bonds into that fatty acid backbone. And we know a lot about these from work done in other plants. So it's a so-called delta 12 desaturase, which introduces this fatty acid double bond to get linoleic acid. And then there's a delta 15 desaturase that takes it on to alpha linolenic acid. And then there's a different desaturase which converts linoleic acid to gamma linolenic acid. And the difference between gamma and alpha, as you'll see on the next slide, is that they occur at different positions on the fatty acid backbone. So this is now a depiction of a fatty acid. This is oleic acid with just one double bond at the 9 carbon position. This is what comes out of the plastid and then gets acted on by these membrane bound desaturases that then introduce another double bond at the 12 carbon position, and you get 18,2, and then you have, as I said, this delta 6 desaturase, which then gives you alpha linolenic acid, and then a delta 15 desaturase, which gives you alpha linolenic acid. So you'll see where we're going with this is that you need to understand and identify those desaturases before you can modify them to in whatever way you want to. And here what we've got is now a developing seed, and these are just this is the embryo of the developing seed, and you can see over a couple of months this embryo matures from torpedo stage to upturn stage to this is filled but not desiccated stage, FND. And what we did was we looked at the transcriptome of these developing seeds, and we identified, using bioinformatics, a whole bunch of candidate desaturase genes. And because there is homology between genes, we then used functionally characterized desaturases from other species, and we identified. So this is the fatty acid desaturase 2A and 2B, and basically these are the desaturase. The nomenclature here is confusing in the literature as well for people not lipidologists. But this naming came from mutants in Arabidopsis, in fact, FAD2, which is the delta 12 desaturase. And so this is the FAD2A and FAD2B. And this is just a heat map showing level of expression in the developing seed. And so you can see immediately that this FAD2A, if we're interested in identifying this delta 12 desaturase, is a really strong candidate because it's got this really high level expression. And similarly, you can identify, for these other desaturases, candidate genes. But of course, they're only candidates until you really prove the gene function. And so this is exactly what we did. And we were then able to then further explore the function of these genes. And we then, rather than just rely on gene expression data and counting the number of transcripts that we come across in gene expression data, this slide comes from quantitative RT-PCR, and you can see, you can see that for this delta 12 desaturase that we looked at the expression of 2 of them, and this one is very impressive in terms of a potential candidate for developing seeds, because it shows this massive expression at this filled, not desiccated stage, when the fatty acids are being made and the oil is accumulating in the seed. And then this is the mature seed. This is young leaf and mature leaf. So of course, when you're interested in finding mutations that will alter seed development or in any kind of way, you want to find genes that are only expressed in the seeds, because you don't want to have an effect elsewhere in the plant. And in fact, you could see then this second gene is very, very lowly expressed. So this gene immediately became our target. And then the other thing we did is that we took the cDNA, the protein encoding region of the gene, and overexpressed that in Saccharomyces cerevisiae, which is brewer's yeast. So this is a really good model for understanding fatty acid functional gene expression, because the wild type brewer's yeast only makes 16.1 and 18.1. So it makes the fatty acids which you find in the plastid of a plant, but not any of those more sophisticated fatty acids. And then when you express that candidate gene, you can see immediately that the level of 16.1 goes down from this level to this level, but you get 16.2 appearing when you express this gene in yeast. And then also the 18.1 goes down and you get an appearance of 18.2. So that's really good evidence that we have indeed got a hemp gene encoding the enzyme which is responsible for desaturating oleic acid. And then we did the same thing with that series of candidate genes for the delta 15 desaturase, and we found a gene which was predominantly, this time, some expression in young leaves and mature leaves, but predominantly expressed and increasing expression during seed development. And then again, what we did this time, because the potential substrate isn't made in yeast, we fed the substrate linoleic acid, and we could see that when we then add that with this vector expressing the gene, that we actually then get we get accumulation of the 18.3 N3. And so and so that's then good evidence, and these different variations of empty vector. We also added GLA and found that we were able to then also desaturate gamma linolenic acid to the steridonic acid, so 18.4N3. So that then, I hope, demonstrates to you that we've indeed got this gene that is producing 18.2, and we've also got this gene which is producing 18.3 from linoleic acid and 18.4 from gamma linolenic acid. And so that then is great evidence that if we could knock out those genes, wouldn't it be really cool? Because then we could basically introduce a block here and introduce a block here, and then we could play around with cross signals as well. So, and that's exactly what we did. And we used this method. This is before we had gene editing going, and also this is now really a combination of, you know, traditional ways to do plant breeding in the 1940s, 1950s, 1960s, where you just used a mutagen and introduced lots and lots of mutations into the genome, and then looked for, often using forward genetics, of course, just screen to get a trait of change in a trait of interest. But in this case, what we're doing is reverse genetics, where we use this so-called tilling approach. I won't get into the nitty-gritty details of it, but essentially this is now using molecular biology, where we can find heteroduplex. We can find heteroduplex formation when we mix a combination of seeds from different individuals. Then if there's a mutation in a gene in one individual, then when we melt that DNA that's mixed from a number of individuals, we can then find a mutation, because first of all, you will get pairing of a mutated gene sequence and a wild type gene sequence to form a heteroduplex. And then we use this enzyme SAL1, which basically cuts DNA wherever you get a heteroduplex. And in fact, this enzyme comes from celery, which is why it's called SAL1. And then basically that allows us then to just using gels, using polyacrylamide gels, in fact, is how we did this. And we found mutations in genes. And the power of this approach as well is, especially when you're working with a dioecious species, such as hemp, in this case Finola, it allows you to identify mutations in the heterozygous state. So even though they're recessive mutations, you can identify them in the heterozygous state, which is almost a prerequisite for us, because it was really, really difficult, still is really, really difficult to self M1 plants. And so basically that's what we did. We did ethyl methane sulfonate mutagenesis, which caused GC to AT transitions. And we generated a mutagenized population. And basically the way that we crossed that, then any mutations we had were going to be heterozygous in the M2 seeds. And then we did screening essentially the way, I'm not going to dwell on it, but that was, once we got the mutagen dose right, this proved really, really productive for us. And in fact, it allowed us to identify this so-called heteroduplex mapping. We got about 60 mutations in 2 gene targets, because the molecular biology then allows you to amplify specific gene targets and do the heteroduplex mapping on those gene targets. And we followed up 15 of those mutations by phenotyping, and five of those showed marked changes of fatty acid profile, which is again quite remarkable. And thus we took those forward into a mutation breeding program. In summary, we had 3 mutations in that delta 12 gene that I showed you that the function of. And I've summarized here what these do. You get these really interesting transitions. We had stop codons, and when you're looking for knockout mutations, you really like stop codons, obviously. But we also got other interesting, this is a proline to leucine transition, and proline, which is a really powerful amino acid in terms of impacting on protein function, because it's responsible for that. Those proline kinks are really powerful mutations as well. And so we got a really nice range of mutations across these 2 genes. And of course, this was described 10 years ago in this Balika et al. Paper, which we published. So then we did experimental This is then the work program that we followed, where we had just gene target discovery and validation. I'm just keeping an eye on the clock here. And then we did lots of screening, as I've just shown you, selected promising mutations, and then we did breeding and backcrossing. And I'm not going to dwell on this because you're all experts in breeding in Cornell and around the world, but basically we took this through. And ultimately, because of course this is a dioecious species, we ultimately end up in crossing, because we could track the gene in the heterozygous state because we had nice molecular markers for these mutations as well. We then ultimately took them through to backcross 4 and then crossed homozygous siblings to get homozygous backcross 4 MH seed. And we then and one of the nice things we were able to do as well in terms of phenotyping is we could just cut off one of the cotyledons, one of the first leaves from the seed, and do fatty acid profiling at the same time as doing molecular markers. And so then just to quickly show you then, I wouldn't be telling you all this if we didn't have really nice phenotypes. When you knock out this delta 15 desaturase, you expect to see an increase in linoleic acid. You would expect it to be a recessive mutation, and that's exactly what you see. The black bars here are the homozygous mutation. You get this elevation in linoleic acid. You get this decrease in 18,3N3 in the homozygous state compared to wild type, and the heterozygote, in fact, is intermediary in a lot of the cases, and that was really nice. And yeah, I don't think I need to say anything more about that impressive phenotype. And what's even more impressive is when you knock out this delta 12 desaturase with either of these two different mutations, which we talked about, which you see here depicted as well. And this stop codon is really nice and powerful. You get nearly a transition from about 10% to almost 80% oleic acid with this single mutation in that gene. And this is what we've focused on. And in fact, we've done field trials and taken this through. And as you had mentioned, Lucia, this is what we registered in the UK as a UK variety. And not surprisingly then, in that high oleic line, there's really high levels of the fatty acid in the triacyl glycerol, the seed oil as well, which is reassuring compared to the wild type where there's nothing. And then really interestingly, things like hexanols, which are, some of those molecules that are give you the rancid smell, that when you look at the Finola wild type seed oil, this is in headspace analysis of the seed oil compared to the high oleic variety, you can see that this is more or less flatlining, whereas these hexanols are really increasing in wild type Finola seed. So just as further evidence of what's happening in terms of volatiles being emitted when you crush the seed. Interestingly, there's elevated levels of tocopherols in the CNAP1HOH, the high oleic variety. And we don't really know why that is, but these antioxidants are present in seed, obviously to protect well, not obviously, but assume because they're protecting the fatty acids from oxidation. And if you don't have polyunsaturated fatty acids in the CNAP1HOH, maybe these tocopherols aren't being used up and there's just more of them. But it's just another interesting, statistically robust trait. And perhaps most interesting is that we do this sort of industry standard ransomat analysis. And if we just focus on the standard hemp and the CNAP1HOH, and this is the projected stability in months of the oil at 3 different temperatures, 20 degrees, 4 degrees, and 0 degrees. You can see this significant increase in the stability. So at 20 degrees, it goes up to 10 months compared to 2 months. And at 4 degrees, it's going from 4 months to 29 months. So you can see, and this is basically getting similar then to olive oil. And similarly, we're now doing a whole range of industry related you know, food standards for this hemp oil, and it behaves very, very similar to olive oil, and so it makes it very, very interesting for a much wider market. We got patent protection of this in the US for that particular allele, and we have plant breeders' rights in New York. We also have been working with partners in Canada, and that's gone into the list of approved cultivars. And I just put this in, which is taken from the Canadian government website, where they record where across all the different states in Canada that the different varieties, there's other varieties as well, but this is Finola, which is one of the most popular varieties in Canada, and it's grown across all these states. And this reflects that in Manitoba, they were trialing CNAP1 HOH in 2023, and they continue to be interested in this. So it performs pretty well. It performs very similarly to Finola, in fact, in the field. So I just wanted to mention, if I can, this is my last trait that I really like, and so this is one of those delta 15. This is one of the other delta 12 desaturase mutations, which we found for some unknown reason. We don't really understand why, but as well as producing what it does is it elevates the levels of gamma-linolenic acid, and you can see here it's not, you know, it's not amazing, but it takes it up to about 7% from 4%, and it may have something to do with the way that this desaturase works. We haven't managed to separate this phenotype from this point mutation in the desaturase, the delta 12 desaturase. And then what we did was we combined that mutation with the delta 15 desaturase mutation, and we've managed to take the levels of gamma-linolenic acid up to about 11%, which, as you know, when I mentioned, is getting much more interesting in terms of a new GLA variety. And we're in the process of trying to get variety registration for this variety, because I think this can be very, very significant commercially as well. It's not borage, as I said, which is 24%, but when you compare the yields that you get for the hemp crop compared to borage, which is a really challenging crop to grow, then per hectare it can be really getting there in terms of production of GLA per hectare compared to borage, if you understand what I'm saying. So this is the range of varieties that I've talked about. I hope I've convinced you that you can really do reverse genetics and get some really nice, interesting new traits out of industrial hemp, and there's plenty more could be done. Of course, you could now use gene editing, but of course, a lot of people still don't want to be doing gene editing, even with the technical challenges, but also the regulatory and public acceptance challenges. So this is still a nice way to go. I'm still a huge advocate for gene editing and GMOs, and I would use it every time if I had a choice, but just pointing out that this route is still very, very broad public acceptance all over the world that we've been using. So that's it. These are the people. Thilo has really led this for me. Monica was the postdoc at the time who was really on this project, along with these other wonderful people. We've had funding from EU and from the Garfield Weston Foundation charity, and from other bits and pieces that I've got over the years. And so I think with that, I'm very happy to stop and take questions. I think I've just been running about 40 minutes. So there, I'll stop sharing.
Perfect. Thank you, Ian. We have some questions in our Q&A chat. I'm sure we're going to have a couple more coming up.
Yeah. Okay, so the first question I see is, do you think we can increase the amount of oil apart from modifying the proportions of the fatty acids? Very good question. Yes. there's different ways you can increase the amount of oil. Typically, if you just consider, the amount of oil as yield per hectare, you can increase the amount of seed, you can increase the size of the seed. I think with oil crops, that ratio of oil to protein in the seed is much more fixed and harder to do anything with. So the increasing the amount of oil is, I think, in terms of, and what we're doing, in fact, is now looking at more conventional breeding programs to increase oil yield. And there are other varieties, for example, that have got larger seed size than the Finola variety, and so there's various ways you can go about doing that. I hope that answers that question. And these are all anonymous attendees. Can change in fatty acid composition have negative effects on seed germination? No, is the clear answer. I've done quite a bit on this in the past as well. Because when I started my lab, the first thing I worked on was seed germination and fatty acid composition. And during the 90s, in fact, there was a really interesting program that was funded by a company worked on by a company called Calgene in California, and then by Monsanto, where they actually made C12 lauric acid in Arabidopsis and in canola. And that's a really, really different fatty acid. And there were some challenges with that in terms of using it, because basically what happens when you germinate seeds is, of course, the fatty acids are converted into sugars and used to fuel growth. But with all of these fatty acids, the enzymes involved in fatty acid beta oxidation can cope with all of these different fatty acids quite easily. And so that conversion to sugar is not inhibited by, and we don't see any impact of any of these on seed germination either. And Daniela, that variety you registered, where is it being grown and how much of it is being grown? So basically, I showed you very quickly there, we're really it's just getting traction now, and I showed you that screenshot from that government website in Canada. So it grows well in Canada. It grows well up there in those higher latitudes, maybe even further north than upstate New York. So that's my bad sense of humor there coming out. But yeah, so it's at that stage where we're really interested. I think it's the trait. I think it's got huge potential, because this trait of high oleic acid, it's now been introduced into soybean, sunflower, corn, and canola, and industry love it. The food industry love the trait because you can fry with it and use it in food processing. I think hemp oil has got all their advantages. It's got a really nice nutty flavor, and it's really good for low impact farming. And as I say, my ambition is to really use this trait to get much better adoption of industrial hemp by farmers as well. So yeah, so I hope that answers your question, Daniela. Is oil, and then another anonymous attendee, is oil from this variety being sold in the UK? Not yet, but I hope it will be soon. Are there seed oil traits or compositions that could be used as proxies for domestication syndrome across different hemp or cannabis varieties? Sorry, I'm now, because I'm not really a plant breeder. I'm just pretending I'm one. Well, what on earth is domestication syndrome? Can you tell me, Lucia? You're a plant breeder.
A exam question. I think it's referring to all those different traits. For example, that they lose or they shatter less, or that they
yes, okay.
produce.
Okay, so all the other traits that you need to facilitate domestication, I don't think so, because pod shattering or seed dropping and dormancy are all regulated by different genes. These genes are really, really, really specific to fatty acid composition. Oh gosh, what would be the best fatty acid profile for sustainable aviation fuel? That's probably yeah. I won't start making any jokes about who might be asking that question. But so okay, the best fatty acid composition for sustainable aviation fuel is more like C8 and C10 fatty acids, so much shorter chain fatty acids than these. And I really wouldn't go near an aeroplane with polyunsaturated fatty acids because the whole aviation fuel, the oxygen is their, and desaturation is their enemy. So yeah. So yeah, so see, and people, I think that I mentioned lauric acid, C12 and C10, that was played around with, and anyway, you would want really, really high yielding crop to give you aviation fuel and be competitive. Whereas with industrial hemp, you're looking at more at a higher value end of the market than aviation fuel. Did you observe any change in the protein concentration? No. And the either concentration or composition, and as I mentioned, because the fatty acid composition of hemp protein is very interesting as well. But is it commercially available? Not yet. We're in the process of commercializing. Yeah, Daniela isn't giving up here. So speak the role of screening genetically diverse germplasm for unusual oil profiles. Good. So this Zachary is asking about looking at germplasm for unusual oil profiles. Very good question, Zachary. We haven't done much of that because we haven't really had access to a huge germplasm, and to be honest, there isn't really a huge diverse germplasm of industrial hemp varieties. And we've looked at a few, but we haven't seen anything come out that gives you a different fatty acid composition in the seed. We haven't spotted anything, in fact, that's different. It doesn't mean it's not there. And then the last question from Joy Beckerman: any general claims that are made re hemp seed oil, the most prevalent being that there is a perfect ratio of omega-3 to 6. How accurate is such a statement, and if one should apply a qualification to it, e.g., generally speaking, most varieties provide a perfect ratio of omega-3 to 6. What is the most appropriate? Oh, good question, Joy. I'm not a nutritionist, and I would never assume that I was qualified to state what's the ideal omega-3 to 6 composition, because I think it depends on who you're speaking to a significant extent. All I will say is that that ratio that you see in hemp, in traditional hemp, if I would call it, and all of the varieties that are out there, it's pretty much fixed with that low amount of GLA and much higher amount of ALA, because you could see that GLA and stearidonic acid are very low, 4 or 5%, and ALA is much higher. So there are applications where people like that ratio of omega-6 to omega-3, but of course there is also an industry need for ALA on its own or GLA on its own, and that's why if I could go back, that's why I'm quite excited about the prospect of developing a variety which is not making ALA, but is making significant amounts of GLA, because when you take those two oils and try and separate them in the factory, it's quite difficult to separate ALA and GLA. Whereas if you produce a variety which is just making GLA then and linoleic acid, then for the processing of that oil, it's much easier to get pure GLA out. So and so industry often have requirements for that downstream processing that are interesting targets as well. So it's not just the end user targets. So I'm not trivializing the question at all. I think there's, it's a very good question, and it depends on who you're talking to and what nutritionist you're talking to. Okay. How about increasing lysine from Andrea Herman? Yeah, like I haven't really focused on that. All I know is that what I've read is that the ratio was pretty good. It's another job to increase lysine. Increasing compounds like that, you really need to upregulate biosynthesis of them, and that becomes a whole other challenge where you really do need to start thinking about doing. Well, you could forward screen and see if you could do it, but upregulating genes by gene editing or GMO would be the way to go if you want to increase lysine.
Awesome. I think we got to the end of the list, the questions. Thank you so much, Ian, for your fantastic talk, and thank you for answering all our questions. For
pleasure.
Everyone, yeah, we've recorded the talk. We're going to upload it to our SIPS YouTube channel in the coming days. Thank you so much for connecting today, and see you in 2 weeks.
Thank you. Bye-bye.
Bye, Ian. Have a nice rest of your week.
This has been a production of Cornell University, on the web at cornell.edu.